Bueno, entonces vamos a comenzar con la presentación de nuestro ponente del día de hoy, que es el doctor Joseph Cook, José Cook para los amigos y colegas. Entonces, el doctor Cook creció justo a unos kilómetros al norte de la ciudad de Chihuahua, en Silver City, en Nuevo México. Es profesor distinguido en biología y curador de mamíferos en el Museum of Southwestern Biology en la Universidad de Nuevo México, en Albuquerque, del que también fue director. Anteriormente fue profesor y curador de la Universidad de Alaska en el periodo 1990-2001 y director de biología en la Universidad Estatal de Idaho en el periodo 2000 a 2003. La investigación que realiza Joe se centra en la conservación, en evolución molecular y sistemática de mamíferos, en la que las expediciones en campo de su grupo de trabajo son un componente muy importante y con los cuales ha creado grandes colecciones de hospedero parásito para el neotrópico y para la región de Beringia. Joe ha sido ganador de la beca Fulbright en Uruguay y Ecuador, de la beca del Club de Rotarios en Bolivia, la beca Sitka Sound Science Center en Alaska, y ha sido distinguido con el premio Joseph Grinnell de la Asociación Norteamericana de Mastozoología. Él fue presidente de la Alianza de Colecciones Científicas Naturales en el periodo 2016-2017 y presidente de la Red de Coordinación de Investigación AIM-UP, las cuales integran recursos digitales basados en información de colecciones para iniciativas de educación. Entonces, el doctor Cook nos va a presentar el día de hoy la charla titulada Construcción de infraestructura crítica para garantizar un futuro sólido para la mastozoología. Entonces, muchas gracias, Joe. Thank you very much, Joe, for being here in Chihuahua.
Gracias a Jesús. Gracias a Enrique, Cristina y muchos más para este honor para mí dar una charla sobre este tema. Su sociedad está muy fuerte, es impresionante. Teria, por ejemplo, es una revista, una journal muy fuerte ahora, gracias a Tiku por eso. Ayer fue charlas muy buenas sobre musarañas, sobre osas, sobre howler monkeys, y es muy para mí a ver la ciencia acá en México en este momento es muy bueno. Y gracias por esta oportunidad a presentar algunas ideas sobre museos y su rol en ciencia. Y voy a presentar en inglés hoy. Si no presentar en inglés, esta charla dura más que 1 hora y media. Pero mis positivos están en español, y si tiene preguntas y si no hay algún que no entiendes, por favor, páreme y pregunta, como una discusión. Anoche también fue fantástico, los tacos, por supuesto, de Chihuahua, y también la boca, ¿no? Órale. Y ahora está el show para Gaby. Gaby es su show ahora. Ok, entonces. Okay, important questions for society, important questions for the world right now relate to change. if you look at these questions that society is asking scientists to help them address, these questions are questions like climate change, right? We hear a lot about that now. Changing habitat conversion, pollutants in the environment, food security, the introduction of exotic plants and animals around the world, loss of biodiversity, and most recently, emerging pathogens. All of these themes, all of these are questions of change. And today I want to try to make the case that museums have an important role to play in addressing those questions. All of those questions, in fact. And biocollections have the ability to establish these baselines or baseline conditions that allow us to evaluate change over time. So our planet is changing. How do we know that it is changing? We have to be able to go back in time to understand what conditions were in the decades of the 1960s, 1970s, 1980s, 1990s, and so forth and so on. How do we get that information? Biocollections are one of the only places for biodiversity that we can go to get specimens that were collected in those decades that we can then use these new technologies that we have, great technologies, to address changing conditions. But we have to establish the collections in a very powerful way if they're going to be useful to address these questions. So we have to grow the collections. So the big theme of my talk today is how do we grow our natural history collections, in this case for mammals, and all the associated things that live on mammals, the parasites, the symbionts, so that we can understand how conditions for mammals and conditions on our planet are changing over time. Okay, so just an outline for the talk. Please do not fall asleep in this talk because it's a little complicated, and if you fall asleep now, you'll be really lost later on. I'm going to try to tie together different themes through this talk and give you just a little picture of some of the work that we've been doing at high latitudes in Alaska, and Siberia, and Mongolia, and Canada, as an example of one way that we can grow our collections. And so a little touch about our research. I'll first start, though, with just giving us the concept of what a holistic specimen is today. And another point that I want to make is museums have to really think their role in science. They have to rethink their role in science, because it's very different than the role was 20 or 30 or 40 years ago. We still do systematics. We still establish taxonomy. That's an important part. But now we are doing many other things with our collections that we were not doing 30 years ago, 40, even 20 years ago. So I'll talk a little bit about our field work, and I'll talk about other ways that museums can get specimens, and then we're going to focus on emerging pathogens because that's a very hot topic right now, and a very important one, and then we'll talk about how to build this infrastructure for biodiversity. Okay, so what is a holistic specimen? This is a very big deal in museums right now. We hear a lot about the extended specimen. And I want to put the extended specimen in the perspective of the holistic specimen. So there was a book published about 3 years ago by a number of ornithologists making the case for how specimens are now being extended in terms of the uses that are being made. But to guarantee an extended specimen, you first have to collect a holistic specimen. And what is a holistic specimen? For mammals, this is very different than the way we collected specimens 30 or 40 years ago. Now, in addition to the traditional skin and skeleton that you see in the center there on the left, we also take metadata, which we've taken for a long time, like measurements. We take an exact location on the planet, a GIS. We also take a date, so we have a time stamp on every specimen. And now we also collect tissues, frozen tissues, multiple kinds of tissues that are used in a variety of different studies. And we try to collect all of these symbionts, all of the ecto and endoparasites from every specimen. So that with every specimen, every mammal that we collect now, we collect a complete community of things that are living on the specimen. Each of these specimens, then, if we move to the right, each of those specimens that can then play into this concept of the extended specimen. So after the specimen is collected, it may get used in many different kinds of studies, and each of those studies generate a lot of new information. And all of that new information, whether it's done by a physiologist, or a geneticist, or a virologist, or a chemist, maybe an isotope biologist, all of that information gets tied back together through that original specimen. And so the specimen becomes a really important integrator across many different disciplines in science. Because the specimen was collected with a GPS unit, it has a location on the planet. Now, all of that information that is generated through the extended specimen can now be brought into GIS applications. These become the biodiversity layers for GIS, both through time and through space. And so they form a really important role as integrating across different disciplines of science. And you can see the many different kinds of science that take place there. So what museums are doing these days is not only are they collecting many different kinds of samples, but they are now integrating many different kinds of data. And so databases and informatics has become a major role for museums, keeping track of other scientists' data. And so our data, our specimens, for example, are tied directly to GenBank, and you can toggle directly from GenBank into our collection, or from our collection into GenBank, or from our collection into IsoBank, which is the new database, data repository, like GenBank, for isotopic ecologists. And so we're building lots of big data. 3D CT scans, computed tomography scans, CT scans, generate tremendous amounts of data. All of that data needs to be kept and tied to the original specimen so that we can keep track of all of these big data. Is there a bird here? It's tricky. Oops. Okay, so the idea of the holistic specimen is that you have microbiomes and parasites and viruses and isotopic ecology and molecular ecology and genomics and metagenomics and transcriptomics and proteomics. All of these get tied together. So you have, for example, pathogen X that occurs in Tejido or in tissue Y on an individual mammal specimen Z, and this corresponds to sequence and DNA sequence, and maybe an expression profile, and maybe an isotopic profile, and it has viruses and microbiomes, D through G, and parasites H through M, and actually for the parasites and the microbiomes, you can do all of those same things, and so pretty soon you're developing a very integrated view of the biology of this specimen and the environment that that specimen was in at a particular location on the planet at a particular time. A very powerful view of the biology. Okay, so what is the problem? The problem is that our collections in the United States, and we have one from Canada here, are not growing today. And so the largest museum that we have for mammals is the Smithsonian. It's the largest on the planet, at over 600,000 specimens. These data are a little bit, these are from, well, at about 600,000 specimens. 590,000 is what it shows there. So the annual growth in the Smithsonian is about 1,000 specimens that they are adding per year. The annual growth from the American Museum, which is another very large museum, has been less than 200 specimens per year over the last decade. And these rates of growth are very, very low, right? The Smithsonian, if it's about 600,000 specimens now, it would have taken 600 years to accumulate that size. And so we're not seeing a lot of growth in the major museums today. And so we're not establishing the infrastructure that we need during a period of the greatest change on the planet. Right now, mammals are changing radically. We're losing all kinds of populations. The recent estimates are 70% loss in recent decades across mammals. In some cases, we're losing species, but certainly we're losing many habitats, right? And we're not establishing the record that would allow future scientists to investigate what caused these changes or how these changes will play out in the future. Okay, so the case here is that this is a platform for integrating diverse investigations, but only if we build these collections so that they are temporally deep, they're spatially broad, and they're taxonomically diverse. So we have a lot of work ahead of us to build the infrastructure we need for all of these other scientists that use samples. And a lot of scientists use samples these days. So how do we build these collections? Well, one of the ways we build them is through field work, and that's the traditional way that collections get built. And so very quickly, I'm going to step you through a project that we did at high latitudes across North America and into Asia. And so this was a project called the Beringia Coevolution Project. Mammalogists worked with parasitologists. We went to the field and we developed a large collection so that we could study a series of questions across this region. Next slide. So Beringia is a very dynamic region, and it has been very dynamic over the last 2.5 million years during the Pleistocene, during the Quaternary period, because of glacial advances. And so during these glacial advances, there were connections between North America and Asia. Next slide, please. Actually, in that last slide, it's an interesting picture because there's both polar bears and brown bears that are feeding together there, which is happening more and more now along the coast of northern Alaska. So this area of Beringia is shown here, and what happened is when there were full glacial advances during the ice ages, the sea level dropped about 120 meters, which exposed the land between Asia and North America, between Alaska and Siberia. And so mammals were able to cross back and forth because this region was not glaciated. So as you can see here, the major ice glaciations did not cover Alaska during that period. It was essentially a refugium that allowed mammals to move back and forth. Next slide, please. And so this great land bridge was recognized in 1937 by a botanist, Hultain. He was very fortunate that he recognized it because he was headed to Alaska from Europe, and he took a boat from Japan, and fortunately his boat sunk in Siberia. And so, being a good biologist, he simply got off in Siberia and collected plants before he went to Alaska. And when he got to Alaska, he realized that they were the same plants. And so he thought about that and suggested that they must have been connected, two pieces of land in the past. So these are what glaciers look like in Alaska. We still have these. We are losing them very quickly, as you know, due to global warming. This is near Juneau. Next one. The glaciers form a big barrier for mammals, right? And we had multiple expansions of glaciers, more than 20 of these glaciers that rolled over the Earth, over the northern hemisphere, and all of the mammals had to leave, and then receded, and the mammals came back. So a very dynamic movement of expansion and contraction back and forth. Next one. And so we can ask this question, what role did Beringia serve in terms of a crossroads between these continents, and what role did it serve as a refugium for diversification, as a cradle for new species? Next. And so this is some of the hypotheses we had with regard to how species moved back and forth across this very dynamic region. As they moved back and forth, they should have formed some contact zones. Those are the hash marks where species collided, came back into contact. And so there should have been areas where there were contact, where species came back into connection with each other. And so we proposed those, and then we went out and studied them using DNA. Next slide, please. So the field work entailed a lot of planes and boats and rafts because it's very difficult to move around this region. It's logistically very, very tough. And we collected specimens across this region over a period of about 25, 30 years. Next one. And so this is an example of the sites that we visited in our collections. And during that period, from 1999 to last year, we collected about 53,000 mammals. Now our field work only produced about 10,000 of those specimens. The other 40,000 came by working with local communities and working with natural resource agencies and wildlife agencies to collect salvage specimens. These are carcasses that come from commercial trappers, from hunters, from a variety of different sources that provided specimens. So we were able to get a lot of information from specimens that otherwise would just be thrown away, right? That would go to a dumpster. So many of the locations, you can break this down over different time periods so that we are trying to build collections that are temporally deep. And so you can see over these different 5-year time periods where our collections were. We tried to revisit some sites so that we could look at change over time. If you look at the bottom there, you can see that they are rodents on the top. Rodents were the most commonly collected species for us, but lagomorphs and shrews and carnivores we got a lot from trappers and so on and so forth, right? So a lot of different specimens, and then across the bottom are different countries that these specimens came from. Next slide, please. So an example of one of the studies that was done, and most of these studies were done by graduate students. Of course, they do all of the work, right? This is the example of Sorex cinereus. This is Andrew Hope's work, where he looked at this complex Sorex cinereus that is thought to be perhaps 13 different species with distributions like this across North America, including down into Mexico and over into Siberia. Next slide, please. And he built evolutionary trees, which is the kind of analyses that we do in my lab, to try and understand the relationships among those different populations across the space, to then understand the dynamics of movements. Not only using evolutionary trees, but there's a lot of other statistics we can use these days that tell us about expansion and contraction, right? And so there's signals in the DNA about expansion and contraction that allows us to understand not only did this population recently arrive here or did it persist for a long time, but also when did that happen? When did it expand? And so we get very good signals of time, particularly as we add additional genes, right? And so most of this work started out in the 90s with a single gene, and now the study is 5 genes that was done in 2012, and now we're doing full genomes, which are much, much more powerful because they give us this nuanced view of expansion and contraction and also contact, hybridization. Next one, please. So for this, this complex of shrews, there was a major division about 170,000 years ago that split the shrews into basically a xeric form and a mesic form that was south of the ice sheets in North America. Next one. And then about 28,000 years ago, the xeric form, which had expanded into Alaska, was left in Siberia. And so there was a connection, a movement from North America into Siberia that took place at that time. And so that's the way we use geology and the geologic history and our DNA to try and understand the diversification of events that took place over time. Next one. And so slowly we're putting together, this is many, many different labs. There are birds on here and plants on here, many different scientists that are trying to piece together the history of colonization back and forth between Asia and North America. Not only of the birds and the mammals and the plants, but also all of their parasites, so that we can begin to understand the evolutionary or the coevolutionary history of hosts and parasites. Next one, please. Okay, so sort of in summary of that work, the ice ages fragmented populations and promoted diversification. This bridge was a crossroads for many, many different species. We see that deep in time, there were some species that made it to North America or went from North America to Asia. Actually, most species moved from Asia to North America. That's the predominant direction. But deep in time, things like Sorex did that, and then we didn't see another expansion into North America until very recently. And so there was a deep expansion, and then diversification independent in North America, and then just very recently we see some connections across North America and Asia. Beringia was also a cradle for diversification, so that this refugium produced new species during that time period, things like ground squirrels, for example. And there's significant phylogeographic structure as we look across these species, and often the boundaries of that structure coincide across species. So this is the area of comparative phylogeography. And so we often see at the edges of the ranges of many species, the edges of Beringia, for example, or the edges of ranges for many species, we see contact zones where different forms are coming together. And the same is true for the parasites. We're just getting into the parasites. The biggest news on the parasites is there's a lot more species than we originally thought. So one tapeworm, for example, very common tapeworm has now become 22 species, right? Because the morphology on tapeworms is very difficult to identify species. Okay, so if we take from the publications that have come from this work to date, and just take the key words from those publications, and we build one of these spaghettigrams, what you can begin to see is the integration that can take place across different studies, across different taxa, across different regions. So specimens begin to integrate across many different themes as we build this infrastructure, and many different kinds of scientists begin to use this work. So you can see many different mammals on there, but you see parasites, and you see ecology, and you see evolution and phylogeography and climate change and so on and so forth. Different keywords that pop up across all of these different studies, and all of these studies are immediately tied together through these specimens. Next one. Okay, so in the near future, there's a lot happening in the Arctic, because there's a lot happening everywhere, right? But in the Arctic, temperature has been 4 times, changes have been 4 times faster. 4 times warming rate in the Arctic over the average on the planet. And so things are happening very fast. And so now what we're trying to do is use new techniques, spatial techniques, like GIS, and combining this with genes. And we saw talks about that yesterday here at this meeting. We're able, because we're using genomes now, to revise our understanding of rates of evolution and the timing of these events. And then we're beginning to synthesize it across many different species to understand the general processes related to diversification in this region. And all of that can happen because we are building these big collections. Okay, but there's other ways to build collections, right? You can collaborate with natural resource agencies. Many natural resource agencies have freezers. They come in contact with dead animals on the road, or they come in contact with dead animals all the time. And these could become important specimens if we can build collaborations or connections with these agencies in Mexico or wherever it might be. Protected areas the same way. There are agencies that work with animals that cause damage, like beaver and other species. In New Mexico, more than 8,000 coyotes are killed every year by our federal agency that helps ranchers, right? And those coyotes should go to a collection because they're important. They should not go just to the dumpster. So there's many ways to build specimens. Veterinarians are another source. Trappers, hunters, and farmers, as well as virologists. And virologists have been a very big source of specimens for us. Next one. We know a lot about pandemics now. The world does, right? This is from yesterday from the Johns Hopkins website, where it shows that 6.5, almost 6.6 million people have now died because of this pandemic. Tremendous cost to the economy. In the U.S. alone, it's estimated at about $16 trillion, right? So when we talk about building collections, and collections being super important for virology, and we'll talk about that in a second, the cost of building these collections is tiny, is very, very small when compared to the cost of dealing with the pandemic. And the problem with the pandemic is that our approach to emerging pathogens is always reactive. We always react after it has happened, rather than trying to think about forecasting or predicting where these pathogens may come from before. And museum collections are a really good place to go to screen for pathogens, to then understand, using GIS and the distribution of a particular host, where particular pathogens may pop up. But we haven't done that. We haven't built that biodiversity infrastructure that would allow virologists to screen for new pathogens, to allow us to predict or forecast. Next one. So I'm going to use the example of hantavirus because it's a pretty good example of how collections may be used. This is actually a slide, the only slide I could find in Spanish, about hantavirus from Peru. But basically, you've all heard about hantavirus, and what we knew about hantavirus up until let's say 15 years ago was that hantavirus was carried by rodents, and this was an important paradigm because it was first identified in Korea during the Korean War, Hantan region of Korea. That's where the name came from. It was not known in the Americas until the 1990s in any of the Americas. Next slide. And there was one strain that was identified in Maryland in the United States before Sin Nombre in 1993. I think the Maryland strain was in the early 90s. It didn't cause any damage to humans. Nobody was worried about it. But then Sin Nombre killed 10 people in the Four Corners region here in my state in 1993. And these were young people. These were young, healthy people, mostly on the Navajo reservation. And they didn't know what this was. These people were dying. They couldn't figure out what it was. It happened very, very quickly. Next slide. And the healthcare system fell apart in New Mexico. This was the sign that was posted on the clinics. They said, stay in your car, do not come into the clinic, right? And that is not what you want in healthcare, a healthcare response. But we saw the same thing with COVID in many cases, right? Because we didn't know what was happening. Next slide. Because we had collections, the CDC was able to come to our tissue collections and quickly determine that it was the deer mouse that carried this virus. Okay? They found the virus, they didn't know where it was coming from, they came to the biodiversity collections, and they were able to tell it was the deer mouse, right? And so this initiated a number of studies. Next slide. And to date, in New Mexico, there have been over 100 deaths. In Colorado, the same way, from hantavirus. This is recent data from the CDC. Next slide. The Sin Nombre virus in New Mexico was the second hantavirus described. This is what we know today for hantaviruses in the Americas. Scientists got busy, virologists, they mostly used biological collections, and they were able to identify many, many forms of hantavirus, including Andean virus, which occurs down in Chile, in Argentina, which has a much higher mortality rate than sin nombre. The sin nombre in New Mexico, about 25% of the people die that get sin nombre. In Argentina and Chile, about 65% of the people that get Andean virus die, right? So it's much more pathogenic. Okay, so these were the new hantaviruses, and again, we were working under this idea that hantaviruses were only in rodents. But then we began to look at other mammals, next slide, and we discovered that moles and shrews have hantavirus, and most recently bats. And of course, if bats have hantavirus, because bats often live with humans, this is a big deal. Next slide, please. And so this is sort of what we know now about hantaviruses across the planet, across rodents, which are in gray, and shrews, which are in red, and bats, which are in blue. And you'll notice for bats, we still have no really good record in the Americas of a bat-borne hantavirus. It's been found in a bat, but it was a rodent hantavirus that was found that had spilled over into the bat. But bats likely have hantavirus in the Americas. We just have not done the hard work yet of screening the samples. And so the work is ahead of us to build that infrastructure that will allow us to understand the distribution of the viruses. Next one. When we look at this hantavirus from an evolutionary standpoint, as it turns out, they have a very deep evolutionary history with mammals. And so they tend to form the viruses, clades, within particular hosts, rodents, or bats, or shrews, or moles, across continents. So that suggests a very, very deep evolutionary, co-evolutionary history of this virus with the mammals, although there are many examples of spillover. Next one. Okay, so now on to One Health. Let me see how I'm doing for time. We've all heard of One Health. There is another thing that is talked about, which is eco-health. So these are two big movements. They came from a little bit different backgrounds. Eco-health came from ecology, evolution, and conservation. One Health came from medicine and veterinarians and parasitologists. But they both have the same conclusion, and that is to solve the question of emerging pathogens, you have to take a very integrated approach, right? And I would say that both of these, unfortunately, have missed collaborating with important biodiversity infrastructure, which is museums. And so we're trying to change that conversation now so that they understand that museums have an important role to play within One Health approaches to emerging pathogens. Next one. And that's because specimens, particularly if they're holistic specimens with tissues, can provide us a lot of important information about pathogens. Sort of baseline environmental conditions. They can help us rapidly identify pathogens like we did with hantavirus. They can provide our ability to survey over time, to remain vigilant over time, to see if things are changing, emerging. They can tell us about the interactions between pathogens and other components of the microbiome, right? So we're just beginning to understand how these microbiomes work. And so what are the interactions between these pathogens, these viruses, and other parts of the microbiome, or even things like fungi, right? In the lung, there are many different species of fungi, and there are viruses. What are the interactions? What are taking place? We can get that from frozen lung samples from museum collections, but only if we build that infrastructure, that library of diversity. They give us, again, collections give us this time and space aspect that's so important, and integration. Okay, so just in this example of another way to build collections, we've been working with the Gorgas Institute in Panama for the last 20 years, ever since hantavirus popped up in Panama, and actually closed their Mardi Gras, shut it down in the year 2000. And so they came up and we worked together, And this is a biomedical group. This is a bunch of doctors. But we convinced them that it was important because they were killing lots of mammals to survey them for viruses, that they save all of those mammals. And so over time, we've worked with them to build a big biorepository for them. Next one. And so this is the locations in Panama where we've sampled. And today, nearly 11,000 mammals, actually more than that now because we haven't been down for a couple of years because of COVID, more than 11,000 mammals have been archived that now provide very nice time and space dimensions for understanding change in hantavirus, but not only hantavirus. Time and space change in each of these mammals. If you're not even interested in viruses, you can now learn about these mammals using stable isotopes or genetics or 3D CT scans, whatever it might be you want to use, because we have saved the samples. Next one. We've also worked with a number of other projects globally. So you can see in the third column there, this is the number of specimens that have come from different countries working with virologists. Working with these virologists that otherwise would just throw the specimens away. And that is the typical way that virologists have worked for a long time, is that they're not interested in the host, they're interested only in the virus, right? And screening for the virus. But really we want the integrated view. We really want to understand how the virus is interacting with the host and with everything else that lives in the host. We want that complete story if we really want to understand the virus. Next one. And so this is an example of a voucher specimen. That was the voucher for chocovirus, which is the hantavirus that's in Panama. And our database is tied to GenBank, and you can go back and forth. And to date, this sequence has been used in more than 100 publications, and it is backed up by this specimen. So this is a key point for virology, and that is, if you don't save a sample when you're doing your virus work, you have nothing you can go back to number one, test whether the first study was even accurate, or to extend that study. Because typically, the early virus studies, and even now, may just get a small piece of the virus, right? The RNA, or the DNA, depending on the virus. But you may want the entire genome, so you want to go back to that original sample to extend the study, right? And so having a sample backing your virus is a very important part of the scientific process of replication and extension. Next one. This is what our database looks like. It's freely available online. Anybody can go in and search it, and you can toggle back and forth to GenBank, or to the specimen, or to any other studies that have been done. And so scientists can quickly pick up information in many different ways. Next one. Okay, so this is this field of informatics, right, which is just, as we all know, is just exploding right now in terms of our ability to investigate new questions. And the idea here is we are, again, trying to move from reacting to emergent pathogens to actually predicting or forecasting, getting ahead of it. Because once we start to react, it becomes very expensive. Okay, so again, these databases are tied into GenBank, Isobank, GBIF, which is a big global biodiversity informatics, Map of Life, Tree of Life. The IUCN uses these for basically for their taxonomy now, and their distribution maps. They pull the data so they can make their maps. Next one. So digital data are important. And now that we have these digital data, museums can combine all of their data globally so that we can actually see what kind of information we have globally for biodiversity, and we can understand where the holes are in our information. That gives us a much better strategy for moving forward, for figuring out where it is that we need to build better infrastructure in the future. So we can be more strategic about the way we build this infrastructure. We also ought to think about what systems do we want to collect, do we want to emphasize, right? Because we have limited human capacity to build these collections. So what systems would be best to focus on? Symbiotic systems, maybe pollinators or seed dispersers, host parasites. Maybe we need to collect entire communities, representatives of entire communities, not just the mammals, but at the same time the plants and the other things that are at a particular location, so that over time we understand change better. Next one. We need to give every museum one of these. And you know, it sounds like a joke, right? These are expensive. How are we going to do that? If we spent $16 trillion in the United States on just one pandemic, this is not a big expense. You can put one of these up and running for $1 million. So we need one of these at least in every country. We especially need them in the most biodiverse countries like Mexico and all the countries in Costa Rica and Latin America, because we need to be saving these samples of biodiversity at this moment. They will be incredibly useful in the future for all kinds of questions. We need to get this message out to the policymakers. We can do this. We can build this kind of infrastructure. Next one. Okay, so again, I want to emphasize that this infrastructure needs to be built in these countries, inside these countries. It should not be built in North America or in Europe. We need to build this infrastructure in these countries, and then we need to build the capacity. The thing about museums is they're great places to train scientists, so we can build human capacity in these countries, so that we have the biodiversity capacity on the ground that can deal with the big questions that we need to address in the future that society wants us to address, like loss of biodiversity. Okay, so that's the big one, is we have to train and enthuse the next generation. Next one. These are the decades of change. Are we thinking as scientists, are we thinking big enough? We need to think big. Society needs us to think big and to give them big ideas to act on. We have to push it. The way we've done science in the past will not work in the future. Things are changing too fast. There are ways to develop new ways to get specimens, and the biggest thing is we have to improve our message and our ability to communicate with others, with society, with the general public, with other scientists. Last slide. This is an example of one thing that we've started with, which is what we call a community of practice that connects museums and wildlife biologists and ecologists and evolutionary biologists with virologists and pathobiologists. And we meet every 2 weeks for an hour, and we have presentations, and we talk about ways that we can build bridges between these fields that have been very, very different, very, very separate in the past. So building new connections moving forward. Thank you. Please join us.